I'm a little excited, okay? And I'm excited because I like giving this lecture. I love heart development. It was a huge struggle for me for a long time to understand this. So, and there are a lot of good images that kind of help you understand it. So I always like going from broad to specific. So how many of you guys have been to the carnival and... Oh,
is that too much?
Reduced lounge singer time. Okay, thank you. That's a very nice Notre Dame sweater you have there. How many Domers do we have in here? Wow, it's a bunch. Okay. Now give them some love. They won their non-conference already. So anyway, you've been to the carnival and you see a guy juggling balls, okay? And that's sort of how the cardiovascular system starts. You've got all these clusters, and all of a sudden that guy goes and blows up a big long balloon. And you've seen those guys that make dachshunds out of balloons. But he has two, and he puts them together, boom, has one, and starts folding it, and it's a heart, okay? If you learn nothing else today, that's heart development for you right there, okay? But I'm bound by contract to do a little more than that. I'm declaring that there is no conflict of interest in this. I'm not sure how I could have a conflict of interest here. We're going to do this according to objectives. The first three objectives are going to talk about or be focused on how the heart is developed. And then we're going to spend a couple of moments, 4, 5, and 6, talking about prenatal blood flow, postnatal blood flow, and then identifying the fates of things that change at birth. And then finally, on 7, I have some specific heart defects to take a look at. These are the heart defects that show up on boards. And they're the ones you're most likely to encounter and have to answer questions about time in and time out. So it's always good to start off with, why are we doing this anyway? Well, the study of the embryology of the heart is going to help you understand the gross anatomy that you'll see once you remove the heart. That's always the truth about embryology in adult form. It also will help explain the developmental defects that you see, and that's a big deal. If you go back to a paper that now we would say is old because it's more than 5 years old, defects are listed here. The atrial ventricular septal defects, that's prevalence of 4.71 in 10,000 births a year. Doesn't sound like much. That's close to 2,000 cases per year. And I can tell you, having been on that side of the fence, that percentage doesn't matter when it happens to someone in your family, okay? But that's a big percentage. And that's why you want to know about these, not just to understand the adult heart, but also to understand how to fix it. Now, I put also some links in here for you. They're probably easily accessed through the PDF version of this. If you want to do some extra reading on the heart development, heart defect literature. The steps that I like to take in explaining and understanding each of these is to ask the 4 basic questions. What's the primordium? And by that, I mean, how does it start? What does it come from? What's the position? Where is it located? What's the shape or the appearance of whatever is developing at this point? And then finally, what processes are used? How does it happen? Is it a dilation? Is it a constriction? Does it involve programmed cell death like apoptosis? Those processes matter as we go through here, and we'll see that very well illustrated. There are some keywords that if you know these keywords, you'll be well on your way to understanding heart development. Well, this is what we want to end up with. A nice heart like this, a nice heart that has a venous inflow from the inferior and superior vena cava, that goes right into the right atrium and is pumped immediately into the right ventricle and off to the lungs for the pulmonary outflow for the exchange of oxygen and CO2 and other things happen in the lungs as well. Blood returns to the heart and through the left atrium gets immediately shunted down to the left ventricle and out through the aortic outflow. Okay, and that's the pathway to go through the heart. And if you just describe the pathway, you'll know a lot of heart anatomy. But how does this happen when you start off with a bunch of red balls that a guy is juggling in a carnival that converts him into a single balloon? You know, he just makes a dachshund out of them. It's a really cool thing. Now, this will show up periodically here in the lecture. It's a timeline that is not linear, but it goes through showing the major developments. And after we do a certain segment, I'll show this, and there'll be an arrow pointing to exactly where this happens, or approximately where it happens. Well, the first key concept, and I've got to thank Tom Shannon for this whole idea of coming up with key concepts. I'd like to take credit for this. In fact, forget what I said about Tom Shannon. I taught him how to do key concepts after I taught Chuck Norris how to fight. Okay? The heart, if you think of yourself as an embryo, your heart starts up here, way up here. It's above, it's the most cranial thing, and it swings down and ends up in the chest. That kind of helps explain how the vagus nerve ends up like it does. But we want to take a look at this. This is an early embryo. It's where we almost always start with this trilaminar embryo. And what happens, these are those red balls at the carnival that a guy's juggling. They're referred to as angiogenic cell clusters, and they're going to coalesce. And when they coalesce, they form a tube. And that tube is going to form on either side of the embryo. You actually have two aortas, and then with that folding, it becomes one. So it's cranial at first, and then it's in the future thoracic cavity anterior to the esophagus. What does it look like? It's a tube once it coalesces, okay? Those M2s, they already heard this last year, so tell them to shut up. Wow. Did you just raise an eyebrow at him and that was it? Okay. Don't mess with that guy. So you have the process is a fusion of cell clusters to form a tube. Okay? Uh-oh. Take it. The hammer's going out now. I have a soft voice. So, oh, and we got the other hammer. Chris, were you making all that noise out there?
It was all me.
Okay, yeah. Did you have a tailwind or a headwind riding in today?
26 mile per hour tailwind.
Okay, so you got here in 5 minutes. Okay, good. I want to bring this up again, showing folding. Folding is a big deal when it happens. The head and tail folds, and then those lateral folds. The head and tail folds, the head fold brings that heart into its thoracic cavity location, and the lateral folds brings these two aortas together for a dorsal aorta. And everything's going to center around that. So this happens right about here. This is about 20.5 days. Okay, but by this time, some people don't even know they're pregnant. This stuff happens so fast. Okay, this developing heart then is a tube, and it's going to dilate to form chambers. It's going to constrict in other areas to start the demarcation process of the different chambers that are in the heart. And it's going to have an outflow system, and it's going to have a venous return system, and it's going to rotate. Okay, so it does all that stuff. It dilates, it constricts, and it rotates. And then it lays back on its side. And you'll see that. So what's the primordium for a primitive heart? We have a lot of primordia here. And this is going to be key for you to know. We're going to have an embryonic structure, and the adult structure. And it's clutch that you know this, okay? You gotta know this. You have to make a list of this and put these off to the side, okay, where they come from. That's critical. It comes up so many times on exams, not only this exam potentially, but step exams, all the way even to your review exams once you have a medical license. I don't know if anybody told you this, but you're going to be taking exams for the rest of your lives. So it's great to be a PhD. We don't have to do that because we pretty much know everything. Okay, what's the primordium here? We have a simple tube that's dilating. That's going to have cardiac muscles that will develop in the wall. And you get all the cardiac muscle stuff from Dr. Al-Ghul. The position is it's right anterior to the developing esophagus and the thorax, and it's a tube. What does it look like? It's a tube. And we have dilation, bending, folding, rotation, all that stuff occurring, and I'll illustrate that for you. So this is what it looks like. This is a developing heart. B on the right is a little more advanced than A. If you notice these terms here, sinus venosus, aortic roots, kind of tells you what's associated with what. You have tubes here, and you have tubes here, and a developing heart here. This is the venous return side of things. So when we were talking about the sinus venosus, these are veins. Veins, by definition, carry blood to the heart, irrespective of oxygen concentration. A vein, by definition, is returning blood to the heart. And we know that any of the veins that are destined for the heart are going to dump their blood into an atrium, right or left, doesn't matter at this point, it's an atrium. And then a ventricle, and then eventually it's going to pump it out, and it's going to pump it out through what is early called the bulbus cordus, but becomes the truncus arteriosus, and arteriosus kind of brings up the idea of an artery, and arteries by definition carry blood away from the heart. So that's the developmental side of things. This is what we look like within that first 3 weeks, and that's how we have our arterial flow. Now, this is describing what's called a unidirectional flow of blood. So it's coming in through the sinus venosus, it's going through atria, back out into ventricles, and up into the bulbus cordus, okay? And then out through the aortic arches. What's missing here? There are no lungs here. Why? We don't need lungs at this point. Mom is doing the breathing, okay? So we don't need that. And what else has changed from the previous diagram? This tube has been bent, okay? This tube has been bent so that the inflow and the outflow is in the same location. And I often tell people, I think there's a rap about this, tubes up, base down. The tubes are, okay, some of you aren't too young. Okay, yo, it's tubes are up, that's venous return and arterial outflow, and the base of the heart is at the bottom. That's how you're always going to orient the heart. And this heart now is oriented properly, but it's a unidirectional blood flow. It just has an atrium, it has a ventricle, and that's it. So we have to change this if we're going to have a proper heart, because someday this baby's going to be born, they'll take the breath of life, and they'll start using their own lungs. And this is, has occurred by this point. The tubular heart has started to dilate, constrict, and fold. And now we need to get to a multi-directional blood flow, okay? And this is what it should look like with the right and left atria and the right and left ventricles. And there's a key concept that we need to come up with now. The first thing we need to do is separate the atria, which is the second floor, from the ventricles, which is the main floor, okay? And this happens through something called endocardial cushions. by just the word. This is a cushion. It's within the heart. Why it's called a cushion, I'm not sure. But it's going to be involved in forming a separation between the atria and the ventricles. And it's also going to be in the location of the two valves between the atria and the ventricles, so it will contribute to the formation of the mitral and tricuspid valves. So how does it happen? We've got endocardial cushions. They're going to develop in an area where the atria and ventricles need to be separated. I'd think of it as a floor for the second floor. And it involves something called neural crest cells, okay? Neural crest. Now, it's kind of a running joke, but it's not really so much a joke because it's almost always true. If you ever see neural crest on a board exam as an answer, just check it, okay? Now, that doesn't really give neural crest the service that it deserves. Neural crest cells are amazing. They do so many things. There are books written about neural crest. In fact, I think we should have a neural crest block, but that's not going to happen. Just to remind you what happens with the development of the spinal cord, okay? We have the ectoderm here in this central area under the induction of the notochord, develops a neural plate which involutes and folds, and as these two waves are approaching each other, the edges of these are called neural crest. And before the neural tube closes, this green area, which gives rise to neural crest, it jettisons the neural crest cells. Somehow, these cells know where to go and what to do, okay? And they're called neural crest because it looks like the crest of a wave. So before this jettisons, or before this tube develops, these guys are going to jettison, okay? And now here's a little caveat. Always remember this. If you have any kind of midline defect in somebody, and that can range from a spina bifida to any cleft on the face, anytime you see a midline problem, start going through all the neural crest stuff. Because you might have problems with dorsal root ganglia, you might have problems with the heart, you might have problems with the kidneys, with the adrenal gland. Those are all related to neural crest cells, and it might be related to failure to close. So anytime you have a midline defect, look deeper, think of neural crest and all the things that neural crest can develop. And here's, again, more material. Now notice that I've used Wikipedia here. Now, when you guys do your research assignments and all, we don't want you giving us Wikipedia references because they're not sufficient, but it's always okay to start off with Wiki, okay? I hope that hasn't been communicated wrongly to you. If you look at the end of Wiki, they usually have the references there, and you can look up references there. That's how I do it, and it works for me. Like I said, being a PhD, I know everything anyway, so hey, you know, they just might as well put Jimmy instead of Wiki. Love, that was supposed to be funny. Oh no, it's reverence. Was that reverence? Okay. So this is what's going to happen. We have these things called endocardial cushions. A right endocardial cushion and a left endocardial cushion, but we need to look at these from underneath. So here's what we have. This is that heart tube, and this is what it's going to look like when we have the two holes which communicate with the right atrium to the right ventricle and the left atrium to the left ventricle. And the way it develops is you have these cushions. There's an anterior and a posterior and two lateral cushions, and they grow toward each other. And eventually, in the center here, they fuse, leaving you with what's called an atrioventricular canal. Okay, and this is what it would look like if you were to look, if you were just kind of sit and look up from the ventricle. That's what it would look like. That doesn't really do it for me. So I've come up with a little way of trying to explain this, which does it for me. Okay, we have this big hole called a heart with atria and ventricles that need to develop, and they need to look like this. We need to have atria that communicate with the ventricles through an atrioventricular canal. Now notice that the atria have not been separated yet, neither had the ventricles. So here's what's going to happen. We've got this heart tube and neural crest cells, which always obey the voice of Dr. Shannon, okay? Dr. Shannon is the man when it comes to this block. And so he inspired me to come up with this diagram. You've got neural crest cells that somehow know that they're to go to endocardial cushions. And so probably Shannon does it. So he tells them to do this, and there they go as a group. I have a lot of free time. They go right to that area, and they migrate into this center area, okay? And then what do? Something magic happens. These cushions grow toward each other, they fuse here, and you have an atrioventricular canal on this side and an atrioventricular canal on this side, okay? And it's all this migration of neural crest that goes down to this area and migrates in, and these cells know what to do at this point. And almost always it goes just right. So we end up with these 4 endocardial cushions that form the separation between the atria and the ventricles. They also contribute to the formation of the mitral and tricuspid valves. Okay? So far, so good. We've got atria up here. We've got ventricles down here. So we've got a little house sitting there, but it's not complete. It will become complete close to the 48th day, but we're in between 33 and 42 days at this point, okay? Now, the times are not so much important for you. I just want to let you know kind of where this is happening. 42 days, that's 6 weeks. 6 weeks. Another key concept has to do now with separating the atria and the ventricles. The ventricles are very simple. The atria are not. They're complicated. And they're complicated for this reason. You don't use your lungs when you're in utero. You use your mother's lungs. And so the blood flow has to take that into account. And we're going to come up with words. Septum means wall. Primum means first. So we have a first wall. We're going to have a second wall. And then ostium means hole. And so we're going to, or foramen, we're going to have a first hole, and then we're going to have a second hole. And I like to envision this like pulling down window shades. Okay, you got one, you got another one you're going to pull down. And you'll see how this works as we work through this. Eventually, you will end up with a foramen ovale, and I'll say much more about that later. But that's how, when you're in utero, blood gets recirculated. Fresh blood from your mother goes into your right atrium, into your left atrium, and left ventricle, and gets pumped out of the aorta. And it does that because it has a foramen ovale, which means oval hole. Okay, it's going to close once you're born. So what do we need? We need a common atrium, of course. We need the roof. We need the endocardial cushions having formed the difference, the floor, so that we have a separation from the ventricles. And we're only interested now in the atria. So we're going to have a vertical line that's going to separate the right and left atria. Now, I'm not saying by the diagram here that they're equal in size. It's just we're going to have a vertical line. That's what you would have for a wall. And I want you to think of it as a one-way doggy door at this point, because blood flows from the right atrium into the left atrium, and then it doesn't flow back because of that one-way doggy door. And you'll see what I mean momentarily. And the process involves growth of cells, of course, to form the septa, and then this programmed cell death or apoptosis that has to occur in order for the foramina to develop, okay? So here we are. We have the right and left atrium. The plus is here because there's a higher blood pressure here that is going to force the blood in this direction. Keep in mind that the septum primum is going to be flexible. That's going to be important for you momentarily. And also that we have a hole here. Now, I don't like this diagram because it looks like there's no hole here, but you can pretend that this is not here because there is a hole. And you don't recognize it as a hole at the beginning because there's just this big space where the two atria are not divided. But as this first septum grows down, it gets down, and all of a sudden you see, oh, it looks like a smaller and smaller hole that's developed that will still allow blood to go from the right to left atrium, and that's actually the foramen primum. If we looked at it from the right, now we're looking at a whole heart on the right side. This is the ventricle here. This whole red area is that first septum. Think of it as a window shade that's been pulled down. And it's being pulled down, and it defines a hole here that is just above this atrioventricular separation. And because it appears to be a hole, it's referred to as the foramen primum. Well, what's going to happen? You pull that shade down all the way to the bottom and shut the blood flow off. Well, that's not what you want. You got to have blood flowing through there. So as this is closing, this area of the septum primum, somehow those cells know that it's time to die. And so you have a region of cell death occurring here. And it's through programmed cell death, and it's going to form a hole. And guess what? It's the septum or the foramen secundum, meaning the second foramen. And this will go ahead and close all the way. So let's look at this again. We have the septum primum, and as it is pulled down, it's actually going to close. And as it closes through programmed cell death, we have a foramen secundum develop. And that all happens in that first septum. But at the same time, another window shade is being pulled down. And that's the second septum. And it's going to actually develop into the definitive wall as it fuses with what was left of the foramen, the septum primum. But it grows down so far, and you get that doggy door so that blood can flow in the direction of this arrow. Let's go back to this diagram, okay? Atria and ventricles are separated through growth from endocardial cushions. That has a lot to do with neural crest cells. And we're going to end up with something like this, a doggy door. And that doggy door is going to let blood flow from the right atrium to the left atrium. So it looks sort of like this. You have a septum that's going to develop. Now that's great if you're an adult, but it's not great if you're in utero. So what has to happen is this. Septum primum goes down so far, leaving an opening called the foramen primum. And then shortly after this, the next septum has to grow down as this is continuing to grow down, and it'll eventually fuse here. Now, what has to happen? If this is going to fuse, you need to find a hole somewhere. You need to make holes. Because if that fuses, then the game's over. So, programmed cell death happens, and you end up with the foramen secundum. And that's cool, because now you've got the whole thing separated and a second foramen here. About this time, this is going to be closed, and that's okay because you have a foramen secundum that allows blood to go from the right atrium to the left atrium. And this septum, that's the second septum, is going to grow down. Doesn't go all the way, kind of overlaps the area here so that you've got that doggy door. Now remember what I said about the foramen primum, or the septum primum, that it's a little more flexible. It doesn't matter so much up here because this part's just going to fuse to the second septum. But this one is going to be pushed over and back each time the heart beats. So that we have something that looks like this and a blood flow pattern that is going to look like this. And so we call this hole, once the second septum has grown down to the point where it is overlapping the rest of the first septum, that first septum can swing open, so we call that opening the foramen ovale. Okay, is that kind of clear as to how that might develop? Yes, sir.
Yeah, so just kind of like in development, are cells like dying as, like that all kind of happens kind of at the same time, right? So like as the foramen, the first the cells dying at the same time, so is there blood flow that flows through like both the cells that are dying and those first
Yeah,
as
yeah.
well as that?
Yeah, it does. It goes through the foramen primum and the foramen secundum. There's a point where they're still both open, and then the foramen primum finally slams shut and all. But you really hit on an important thing, and I wish I had a good analogy for this, but my filters must be on. This is such an incredibly dynamic process going on. It's like, well, it's sort of like what's going on across the street at the hospital. There's a lot of different people that are sick with a lot of different things, a lot of different doctors and nurses and other healthcare workers doing all this stuff at the same time. And at the end of the day, good things will have happened. But it's so hard to keep track of all that stuff. If we were reduced to what's happening at that point, it would be at such a warp speed that even the Trekkies and Star Wars aficionados in here would be frightened. Okay, it's so dynamic, and I want you to appreciate that that dynamic part of it. But I want you to appreciate even more how this develops and the fact that when it goes wrong, it can be fixed. Okay, and so now all this is happening by day 46. This should be pretty well finished. So the key concept is that the atrial septum is very complicated, and it's complicated really for one reason. You have to maintain a steady flow of blood to the rest of the fetus's body. So it has to go into the left atrium, left ventricle, out to the aorta, and then back to mom after that. And it involves all these structures, and importantly, the foramen ovale. So another key concept that I taught Dr. Shannon. The development of this interventricular septum is much less complicated, okay? It's something I can really wrap my head around because it is fairly uncomplicated. And it's basically a growth of cells from the floor up. And this is what it looks like. Here we have the left and right atria, and they're formed. They've got the nice foramen ovale here, the doggy door that flips open. And then we have a right and left ventricle. And basically what happens is it just grows up to where it can meet the inferior portion of the atrial wall, which is in the location of the atrioventricular canals, or the endocardial cushions. And what you'll find is that there's a muscular part, and when you open up the heart and you probe around in here, there'll be a really thin area up here, which is called the membranous part. And I point that out to you because if you're going to have a ventricular septal defect, which is called a VSD, that's most likely where it's going to occur. And that's going to be just a little bit beyond when the two atria are separated. Now it gets real. I just love myself, okay? There's a reason for this, okay? Aorticopulmonary outflow. You've got now a fusion of a tube, okay? And it's nuts. It's nuts because you've got to have part of that going from the left ventricle to the lungs. And you've got to have part of that going from the right ventricle to the lungs. And the other part has to go from the left ventricle to the aorta. But it's one big tube. That's nuts. What are you going to do? Well, whoever developed the development of the heart must have known something about pirouette, okay? And this is how I remember it, okay? I would do it for you, but then I would yak, okay? I'm at that age where I can't turn around without becoming nauseous. And you younger guys in the back, eventually that'll happen to you, okay? It just happens. I don't know why. Doesn't occur with wisdom, okay? But we have to separate pulmonary outflow from aortic outflow. And this just sticks in my mind as how it happens. Okay, we have this truncus arteriosus. And by definition, arteriosus means it's flowing away from the heart. And along a spiraling line, we're going to separate these two. I like to think of it as a pirouette or a spiral staircase. And it is going to, again, involve endocardial cushions and neural crest cells. And the key feature here are the conotruncal ridges, okay? And the conotruncal ridges are represented here by the yellow and the burgundy colors. They grow toward each other, but as they grow toward each other, they spiral around in a pirouette formation, which allows for the separation in this truncus arteriosus and for pulmonary outflow to go to lungs and for aortic outflow to go to systemic circulation. And it's all starting in this area of the membranous portion of the interventricular septum. Okay? So as the septum is growing up, in the truncus arteriosus, which is common to this, it's starting to develop this spiral staircase from the conotruncal ridges that will eventually fuse, and magic, you have these two channels that develop. And that's going to take us up to a little beyond 56 days. So that's 8 weeks. So what happens prenatally? We don't need our lungs. We don't need our livers. So we need to have some bypasses for these organs. And these bypasses would include the ductus venosus, which is going to bypass the liver. A foramen ovale, which allows for shunting of blood from the right atrium to the left atrium, and a ductus arteriosus, which is there to collect the blood that somehow got into the right ventricle and got pumped out, but it goes, instead of into the pulmonary circulation, it shoots up through this ductus arteriosus into the aortic circulation. And this is what it looks like. Got a nice huge heart here. We have the ductus venosus here. The foramen ovale would be situated here. The ligamentum arteriosus, or the ductus arteriosus, is here, shunting blood that manages to get into the right ventricle out and into the systemic circulation because it doesn't need to go into the lungs. The only reason blood needs to leak into the lungs is really for perhaps nutritional support. So you're born, you breathe in, and your ductus venosus becomes a ligamentum venosus. Your foramen ovale becomes a fossa ovalis, and you'll see it as a depression when you look in the right atrium. And the ductus arteriosus becomes the ligamentum arteriosus. And then also the umbilical arteries and vein are going to constrict, but we're going to talk about this in the GI block when we look at the inside of the abdominal wall. So the key concept here is that prenatal circulation avoids unnecessary flow to organs that are not yet needed. And postnatal circulation establishes the necessary flow to the organs that are now needed. And those shunts that were prenatal get closed. And there are actually factors like bradykinin and prostaglandins and things like that. Oxygen tension plays a role as well that tells these structures to constrict and to shut off. So let's look at some selected heart abnormalities. The first of which would be what is commonly referred to as an ASD. And it happens more commonly in females than males, a 2-to-1 ratio. And it allows interatrial shunting of blood, which you don't want to have. There are two types, a septum primum and a septum secundum type defect, or ostium primum and ostium secundum. The ostium primum defects occur close to the tricuspid valve, okay? And because of that, they're a little more complicated to fix. Okay. My grandson, my first grandchild, actually had one of these, so they required an open chest procedure and sewed it all up, and I think they put some stimulator in there because this kid is wild. But he's related to me, so it could be that too. There is an ostium secundum defect, which occurs more in the area of where the foramen secundum was, okay, which means it must have persisted. These actually can be repaired by percutaneous procedures. Then there are ventricular septal defects. They're almost always an isolated defect. But my grandson, who now we call Lucky, in addition to having an ASD, had a VSD here, and that was fixed as well. But usually it's just an isolated defect, and it almost always is in this membranous region. And it allows for mixing of ventricular blood. Again, the problem is you're just not fully oxygenated. Okay, those are ventricular and atrial septal defects. There is another one, this is historically very important. Somewhere you're going to get asked this a lot. Tetralogy of Fallot. Tetralogy must mean that there are 4 things, okay? And you can explain the physiology of this as you look at this. And here's a really key point as you guys go into your professions as doctors, okay? As doctors, you're the most revered person of the healthcare team, and rightly so. Tetralogy of Fallot was first recognized by a nurse on the floor who recognized that, are there any nurses here? Anybody? We often have nurses that come through. They make the best students. A nurse noticed that these babies were blue. They were cyanotic, and there was something going on. And she brought that to the attention of the peds cardiology guys, and they eventually figured this out. Here's the pathophysiology of this. Your pulmonary trunk, which leads from your right ventricle to your lungs, it gets stenotic, okay? There's a defect between the ventricles, so you got mixing of blood. The aorta overrides a bit, and there's a hypertrophied right ventricle. Now, why do you suppose you would have a hypertrophied right ventricle? Raise your hand if you, yeah. Are you raising your hand? No, that's your coffee cup. How many of you guys go to the gym to work out? Okay. So what happens, I'm sure you've noticed, I'm getting pretty ripped. As you lift weights, what happens? Your muscle hypertrophies, okay? As you pump against something that's getting increasingly smaller, that's like lifting weights, and your right side of your heart gets bigger. So that's why you have a hypertrophied right ventricle. And this is the diagram then that looks like the tetralogy of Fallot. Pulmonary stenosis, right heart gets bigger, overriding aorta, and an interventricular septal defect. Remember these 4 things. There's another thing called the transposition of the great vessels. The great vessels are the aorta and the pulmonary trunk, they get transposed. They can actually be fixed. What happens here is you've got a right to left shunting, so you end up with cyanosis. The aorta rises from the right ventricle, the pulmonary trunk arises from the left ventricle, and it's often always associated with an ASD and a VSD and a patent ductus arteriosus. Okay? So it is in some subtle ways similar, yet very different from a tetralogy of Fallot. We also have persistent truncus arteriosus, and when this happens, I mean, it's like, whoa, where does the blood go? And again, you have a right to left shunting, you get cyanosis because you got mixing of blood here. So you have a partial development of the AV septum, and it's always accompanied by a ventricular septal defect, and mixing occurs. So basically, anytime you see a cyanotic child, it's time to call a pediatric cardiologist. So the key concepts have been put there for you. I hope this makes sense. It will help you understand the adult as well, but you guys will go into wards and you'll see kids with heart defects. If any of you are interested in shadowing some of the pediatric cardiologists, shoot me an email, and they're always very happy to have you guys tag along. If they're doing any of these percutaneous procedures, imagine this: you got a hole in your ventricles, okay? And so you're put under, and a catheter is slipped into your vein, usually subclavian, and under fluoroscopy, it's threaded in through the superior vena cava, right atrium, through the tricuspid valve into the right ventricle, and they thread the catheter through the defect. And all of a sudden, they flip a switch, and a little thing that looks like a dime comes out, and they pull it back against the defect. They hit another switch, and another little dime kind of opens up, and it's on the other side of the defect, and they mash them together, and they lock together, and then they pull the catheter out, wake the kid up, boom, just a little injection right here through this. And there's a suite here where that's actually done here. And it's also done for ASDs. I'm sure I'm standing before someone who will do this eventually. Okay, so that's why it's important to know this stuff.